Let's move to the last presentation from Praveen Chitneedi from FBN, and Praveen will talk about eQTL analysis.
Good morning to off-site and online audience. Today I'm going to present my workflow which we developed for detecting eQTLs in BovReg project with the BovReg dataset. And we developed with this workflow using Nextflow based technology. This one. So eQTL's association study identified the genetic variants that affects the gene expression. So in the previous talk from Rodrigo, we found he said that 70% of the heritability due to DNA variants changing the gene expression and RNA splicing is due to cis and trans effects. This is a very recent study. So we are also in the same direction to investigate this. So basically, the eQTLs are categorized into two based on the distance between the SNP and also the gene it affects. If the distance is less than 1 MB, they are called as cis eQTLs. If it is greater than 1 MB, they are called as trans eQTLs. So in addition to that, there are some variations of these cis effects based on the SNP and the associated gene expression. For example, when presence of a particular SNP affects the isoforms by splicing the genes, they are called splicing QTLs. And also another variant is when the presence of a heterozygous SNP, sometimes irrespective of parental origin, presence of a particular allele increases the expression of a gene, and presence of this alternate allele decreases the expression of gene. And these are called allele-specific eQTLs. So to conduct a meaningful, significant association studies, generally it requires millions of genetic variants with thousands of genes, typically measured in hundreds of individuals. So compared to GWAS, the eQTL study also requires many intermediate steps for processing the expression data. So in this context, we need some robust pipeline which can be scalable and also reproducible. So from the talk from Jose yesterday, we found that Nextflow is one of the promising technology to develop a robust bioinformatics pipelines. So we also use this technology to develop our eQTL pipeline. So it's like a brief recap from the Jose's talk. So these are the main features of Nextflow. Like it integrates all the softwares required for a pipeline, and it uses container technologies like Docker, Singularity, and Conda. So the main advantage is these are a single unit of software. So in these containers, we can install all the required tools for a bioinformatics pipeline, and this can be the configurations and also the versions which we installed for an analysis can be maintained, and it can be portable to different server environments. So it can be repeated irrespective of any glitch if you are moving the pipeline from one server to different server. And in addition to that, this Nextflow technology also useful for running different kind of scripts in the same pipeline, and seamlessly it can couple different scripts written in different programming languages like R and Python. So finally, it also the Nextflow allows high performance computing environment to run the analysis, like Amazon Web Service, Google Cloud, and whatever it is. So it is very, so when developing a pipeline using these kind of technologies, it is very robust, especially for eQTL analysis. So here is the flow chart of our eQTL workflow. Actually, we developed this workflow using Nextflow domain-specific language 2, which allows to develop the workflow in a modular format. So we basically divided our workflow into 5 modules, and we called them as scripts. So in the script one, we performed the indexing of reference genome, which generates the star index. As we used star for alignment, we have to do this as an initial step. And after that, using the star index, and also we used BovReg annotation for this analysis. And also after alignment, we performed we generated the expression counts for gene and transcript level, and also for the splicing QTL, we generated the junction files. And in the script 2, we extracted all the imputed genotypes which were imputed to whole genome sequence level. So this is the same data which we used for work package 4 task 1. So this is the same data, and we extracted the corresponding samples which have RNA-seq samples. And in the script 2, we merged all the expression counts from the samples into count matrices for the gene and transcript level, and also for the splicing QTL. And finally, in the script 4, we merged all the input data generated from the RNA-seq expression data, and also from the genotype data. And initially we performed the principal component analysis, both for the genotypes and also for the phenotypes, and then finally perform the QTL analysis with this data. So this workflow is currently now available in BovReg GitHub page. So any interested users can download this workflow, and we also provided some sample example data for the genotypes and also for the phenotypes. So users can download this data as well, and then they can also use this workflow for their own analysis. So recently we also submitted a paper to NIR Genomics and Bioinformatics explaining this workflow, and it is currently under review. And in addition to that, we also performed allele-specific eQTL analysis, which is the workflow is slightly different from the previous workflow. Here we have to mask the reference genome for this analysis, followed by alignment. And then there is a standard GATK workflow to generate GVCF files from the expression data. And finally, using this expression data and also the imputed genotypes to whole genome sequence level are used to generate the allele-specific eQTLs. We use the method mentioned in Pro C. Wilkins et al. For this analysis. So these are the data which we collected from different BovReg partners. So we collected from 6 BovReg partners, the RNA-seq data, and also the corresponding whole genome sequence data, sequence level imputed genotypes. And also we got support from CRG Barcelona Jose Group for developing this workflow. So this data also is a composite data which from different breeds. So here is the brief summary statistics which we obtained for the 6 eQTL analysis. So the most interesting part is using BovReg annotation, we found significantly higher number of transcripts when we compared the number of results from ensemble annotation. So here is one of the overlook of the results for the cis-eQTL in the liver tissue. Here some interesting fact is like we found some significantly higher number of hits when we plot this graph across autosomal genotypes in chromosome 18 and 19 compared to chromosome 1 and 2. So we also perform some comparative studies to check whether our results have some meaning. So we found that we found some hits in GCCNV region, which was predominantly expressed in liver tissue in one paper of eQTL analysis. And also, other than that, using the BovReg annotation, we found some a novel and undescribed non-coding RNA signal in the chromosome 6. And also they found a nice overlap of this peak with the ataxic peaks in our experiments. So in summary, we created a bioinformatics workflow for eQTL detection using Nextflow, and which is currently available in GitHub. And we performed eQTL association study with our BovReg datasets. And with the BovReg annotation, we discovered a significant eQTLs previously unannotated transcripts. And these eQTL studies the results from these eQTL studies were used in subsequent packages. For example, in work package 7 for biological trivial selection. I think Emery will be describing this in the afternoon session. And finally, we will integrate this allele-specific eQTL analysis in our main BovReg eQTL workflow. Thank you for the partners for providing the data, and thank you for your attention.
Thank you very much, Praveen. Questions? Yes, I think that we have a online question. Okay, so the question is, did you look at the overlap between your eQTL results and the ones of the catalog text?
No, currently we are still in the process of collecting the information from the eQTL results, and we have an idea to compare these results with the catalog text, because as far as I know, the catalog text used they generated the genotype from the RNA-seq data, but ours is unique, like we directly generated the genotypes from the whole genome sequence data. So it would be interesting to compare these results, so we are in the direction to compare this.
Okay, thank you.
I might add a little bit to this. We did a comparison very roughly regarding the numbers of hits we found, and the numbers are more or less in the same magnitude, but we didn't have a close look at the exact overlap.
Okay, thank you.